Swayam Prabha. Digital India. Educated India.
Welcome back. Today we will continue discussing on the reaction mechanism of formation of methanol, right. So, from methane we are talking about. Methane is such a great starting material to convert or to be converted into biomass, right. So, methane to methanol is hugely important and very difficult transformation, and we have seen in the last class that these diiron bis-mu-oxo intermediate, where iron is in +4 oxidation state, iron is in +4 oxidation state, and oxide is reacting with methane to give an intermediate, or in this case the first the transition state TS1 CH4OH, where both bond making and bond breaking is happening simultaneously. From there we have seen the intermediate where CH3 and OH are interacting still with each other, and subsequently a concerted pathway where CH3 and OH is intimately involved in the CH3OH molecule formation. This pathway, so called concerted pathway, is little bit energy demanding compared to the bound radical intermediate pathway. So, in the bound radical intermediate pathway we see the exactly same transition state where bond breaking and making is happening simultaneously. From there a distinct radical intermediate involving CH3 dot radical and oxo-hydroxy iron 4 iron 3 intermediate is forming. Now that I would say quite interesting. So, what has happened over here, this one of the iron center is iron 4 plus, another iron center is iron 3 plus. Now the oxo is no longer oxo, it is hydroxide HO minus 1 minus, this is oxide O2 minus, right. So, iron 4 iron 3 mixed valent intermediate is formed along with the formation of the CH3 radical. This is what is IBR intermediate of this, which is really preferred over that you see for IC, intermediate for concerted pathway, okay. Intermediate for the radical pathway is this one. This radical then can react with this OH. Of course, the OH has to orient properly, and of course we will see in a moment that this rearrangement or the electronic reassignment has to be done. Some electron spin has to exchange. Overall we will see that this CH3 and OH, once the OH is properly oriented and ready to react with the radical, HO is HO minus, that means 2 electrons are there. So, 2 electrons over here, 1 electron over there is not going to be a radical mechanism. So, this double electron has to be a single electron by transferring one of them to the iron center, and this is where this iron intermediate will come into the picture, and this OH radical and this CH3 radical will combine with each other, right, to give the CH3OH. Let us look at this pathway, which is the preferred one, in little bit more detail. As you see, the energy levels are not too high, it is achievable, and these reactions are achievable at 20 degrees C at nearly room temperature or less than room temperature at pH 7. So, to be able to convert methane to methanol at room temperature at pH 7, I think is phenomenal, right. This is quite exciting. Such a transformation remained very difficult to do even for the synthetic setup. Enzyme can do it perfectly because everything, all the active site is perfectly. We will look at these steps, how methane is approaching this intermediate Q, how this transition state is forming, how these intermediates are, and how the next transition is forming, and subsequently the overall product formation is happening in the by the DFT studies in a little closed manner. Okay, so this is your methane is approaching the intermediate Q. So you see these two intermediate, these two iron centers are iron 4 plus oxide, and rest of the cycle or rest of the ligands are as it is as in the active site. So you have iron sites, oxo and the methane. This approach or this is set as 0 because this is when it is going to come and start reacting. So this is set as 0 kcal, and we are going to look at this overall hydroxylation of methane to methanol by this non-heme diiron enzyme. Okay. Subsequently, this is the transition state that we discussed, where CH bond breaking and OH bond making is happening. Okay. The electron transfer in this state, electron transfer from this CH bond, electron transfer occurs and determines the barrier height. Here it is 17.9 kcal per mole, which is okay, right, which is achievable at 20 degrees C. So this is the highest energy demanding step, and this is the step why we do see kinetic isotope effect when we displace CH4 with CD4. If these are deuterium, this CH bond breaking becomes further slow, because this is the one which is the most difficult step to carry out. CH bond breaking and OH bond formation, this is the corresponding transition state. CH4 versus CD4 will give a kinetic isotope effect value of nearly 22, as you have seen earlier. Okay. During this process, one of the two iron centers will be reduced to iron 3 as this OH bond is formed. This is the transition state, by the way, not the intermediate. So this is the structure looks like in this transition state. If you move on from there on, what will happen once the OH bond formation is happening, we are looking at the second transition state. This OH is now formed, and it has of course have undergone the electronic rearrangement. Overall, now this OH is ready to bind with CH3, and the corresponding transition state will be looking like this. This transition state is very little uphill from the radical intermediate that we have discussed 2 slides back. So these are the two transition state. Once again, this is the first CH breaking OH bond formation step, transition state, which is the most demanding step, and this transition state is not that very demanding, it is rather easy to form, rather less energy demanding, where OH rebounding with the CH3 radical. So this is of course oxygen rebound or OH rebound step with the CH3 radical, but before that the rearrangement of electron has to happen. Let us look at those rearrangement of electrons. So this is once again from this book, from this reference, from this research paper, where this is the radical bound or bound methyl radical. So CH3 dot H here is carbon, this is the p orbital of carbon, this is one electron over there, CH3 dot, donor orbital is there. Here is the acceptor orbital, this is one way of looking at it, one of the orbital we are looking at, dx2y2 of the iron center, which is having beta spin LUMO. So we will see that in a moment. If you are looking at hydroxide HO minus have 2 electrons, and so the 2 paired electron, one up another down, this doubly occupied intermediate or doubly occupied state then will transfer one of the electron from here to the iron center, right. Let us look at that. So this is not really, this OH minus and this double electron is not going to react with the with this CH3 radical, that very easily this OH also has to form the radical. That means only one electron should be over here, another electron should go somewhere, most likely in this iron center. We will see that in a moment. So what you need to do, you need to overlap, so the rotate this p orbital or rotate overall over here to do the bonding between this d orbital of the iron center, let us say iron 1 center, and subsequently one of the electron from here will be transferred to the LUMO, metal based LUMO, and then you left up with one down spin beta spin, one alpha spin on the OH and the beta spin on the LUMO of the iron center. So what has happened, the alpha spin over here is now reacting with the beta spin of the radical to form the CH3OH bond. So these will form the new bond, and then there is the one electron reduction center from the iron center, making it from iron 4 to iron 3. So this HO rotation, this rotation promotes intramolecular beta electron transfer. So intramolecular beta electron transfer from the oxo lone pair orbital to the metal based LUMO. So this is the metal based LUMO. So oxo lone pair to the metal based LUMO, this transfer is happening. The remaining radicaloid, that is over here, after one electron transfer, the remaining alkaloid alpha electron on the bridging oxo group has the correct spin now to recombine with the beta electron of the sigma bond of the CH3 radical, and these two now will form the sigma bond. So what is essentially happening from here, if you look, this is the CH3 radical, this is the di-iron center, this is the beta spin LUMO of, that means dx2y2 orbital for the iron 1 center. We are not looking at the iron 2 center. If you are looking at OH, 2 electrons over there, it will have to rotate. In any case, this is the 2 electrons. So this is single electron, this is 2 electron, it will have to rotate. One of the electron has to, the beta electron has to be transferred to the iron center, and this is what you see over here, the alpha spin the beta spin and the alpha spin upon rotation and transfer of one electron from this OH to the iron center gives rise to a situation where OH radical is there and the CH3 radical is there. Now these two radical will combine to form the sigma bond. So we have seen how CH4 to CH3 radical and CH3OH bond is forming. So overall this is the diagram where you started, as we have seen, with the bis mu oxo species, so called the Q intermediate, and this is the Q star intermediate. Q intermediate reacting or approaching, methane is approaching towards it, that is set to 0, and this CH bond breaking and OH bond formation is happening. So CH bond breaking and OH bond formation is happening. You are having a transition state right over here. This is the most demanding transition state, 17.9 kcal per mole, where if you have the CD4 you will also get the same thing, but it would be significantly slower, and then therefore you will see that the CH3 radical and OH is now formed. This hydroxide still has to form the OH radical, and that is what is happening over here. OH radical is formed and CH3 radical is over there. So they will combine. This is not that very energy demanding step, and this is when they will combine with each other and will give you the CH3OH bound intermediate that we have seen earlier. So to summarize simply, in this case what we have seen, that a di-iron mu oxo intermediate is approached by CH4, radical abstraction, or you see that CH3 radical formation or OH formation is going on between the iron 2 centers. From there on, electronic rearrangement has to happen to give you the hydroxy radical, and then you also have the CH3 radical ready, they combine to give you the product, that is all, nothing else is there. Okay. So if you are looking at CH4 versus CD4, that we have seen that this is having a high kinetic isotope effect value, nearly 22. Quite surprisingly, if you are changing the methane substrate from methane to ethane, just change of substrate, methane to ethane, and if you are studying this enzyme, you see that there is no kinetic isotope effect value between C2H6 and C2D6. Here CH4 versus CD4, you have a huge difference in the reaction rate, right. Over here C2H6 versus C2D6, which is ethane versus deuterated ethane, there is no difference in the kinetics data or K-obs data. So this is quite amazing, right. For ethane, the rate determining step therefore is not the CH bond activation step. So what we are trying to tell here is the scientist has now figured out quite phenomenal observation, and that is methane and ethane reacts completely differently, or at least the intricacies of the reaction mechanism are different. In case of methane, CH bond activation is the rate determining step. Can you imagine that? And just you change from methane to ethane, CH bond formation is no longer the rate determining step. You do not have a kinetic isotope effect value at all, its kinetic KIE is 1, although the K-obs is same, which is fascinating, I would say. So for methane, hydrogen atom abstraction is rate determining. For C2H6, the binding, the approach, the C2H6 is approaching the Q, the diffusion control is happening. So the binding of the ethane with respect to the Q, intermediate Q, is important. See, this is how nature has evolved in such a way, the enzyme is designed in such a way, overall enzyme pocket, the approach road is such that it just fits methane perfectly. Okay. If you want to fit in ethane, that is also not possible, and this is why one should realize that although nature is really great, but nature is only designed for one substrate, and this is also true why we have such difficulty in synthetic setup in mimicking this chemistry, and more importantly having a broad substrate scope for this sort of active site if we are able to mimic in synthetic setup, because nature can do it for one substrate, but synthetic chemists want every substrate over there. That is not even nature has done it, right. That is going to be even more challenging. That is why synthetic methodology development or catalyst development for a wide variety of substrate is always going to be challenging. One thing perhaps would be better for synthetic chemists to do is to identify one great reaction and try to do these transformation effectively, just what perhaps nature is doing. But synthetic chemists' demands are little different. In any case, C2H6, as we are discussing, the binding is the rate determining step. The bond in C2H6 is weaker. Of course, this is weaker significantly, I would say by nearly 5 to 6 kcal compared to CH4. CH4 was 104 kcal per mole, and C2H4 or ethane CH bond breaking would be something like 104 minus 6, let us say 98 or 98, 99 kcal per mole. So this is also saying that CH bond dissociation is not the rate determining step. That means CH bond dissociation is relatively easy, indirectly saying this is also can be proved by experiment and theory. In any case, the CH bond dissociation is not the rate determining step, but the approach of ethane or diffusion of ethane to the intermediate Q is the rate determining step. But for methane, diffusion was not of a problem, but CH bond breaking was the problem, right. For ethane, you can see that the approach of ethane towards the Q is problematic or the most demanding. Okay. So a number of substrate one can study. One such substrate that has been studied, again this is a different paper, so that is the nitromethane, and in this case actually react IR can be used to monitor these by IR spectroscopy, the kind of the dissociation or the CH bond breaking or the new product formation can be followed by the react IR. So CH3NO2 versus CD3NO2, can be studied by react IR. Of course, one can still think of doing the UV visible spectroscopy, but this is a fantastic way of studying the reaction mechanism. Once again, the CH3NO2 and CD3NO2 will give this sort of plot, K-obs versus nitromethane at pH 7 and 20 degrees C. This also gives rise to a very high kinetic isotope effect value. As you see, between CH3NO2, which is faster, CD3NO2 is significantly slower. Okay. So, this is not by UV visible spectra study, but by IR study. You can follow the kinetics of this reaction with respect to the concentration, different concentration of CH3NO2. The study is done similarly as we were seeing for intermediate Q, and then change the CH3NO2 to CD3NO2 and vary the concentration of CD3NO2, you will get the plot as is over here. Overall, many different substrates are studied, okay, and this remains quite interesting. For methane, as you see, nearly 23, 22, 23, 23.1 plus minus 1.1. This is the kinetic isotope effect value. As you see, that is for CH4 and CD4. The CH bond breaking is the rate limiting, and therefore a huge kinetic isotope effect. For CH3NO2 and CD3NO2, once again there is a large kinetic isotope effect, not as large as CH4 CD4, but CH3NO2 and CD3NO2 is still high kinetic isotope effect. This is showing that once again both for methane and nitromethane, CH bond breaking is the rate limiting step. One can even study the nitrile, let us say acetonitrile, for its hydroxylation chemistry. Acetonitrile versus CD3CN is giving perhaps the highest kinetic isotope effect value in this series, which is once again saying that the CH bond dissociation is problematic step rather than the diffusion, which is happening in the other case. This is the diffusion control process. Now, both for all for methane, acetonitrile and nitromethane, it is a significant kinetic isotope effect value, indicating that these are the substrate wherein CH bond dissociation will be the rate limiting. I think it is also quite fascinating, as we have discussed, that methane to ethane, you change the dimension completely. Methane CH bond was the difficult step, ethane of course CH bond is little weaker, nearly 98, 99 kcal per mole bond dissociation energy, but this is no longer, CH bond dissociation is no longer the rate determining step. The rate determining step is going to be the one wherein it is diffusing or approaching the intermediate Q, and that is the difficult step rather than the CH bond breaking. And such a similar case will be happening in case of methanol. Methanol versus deuterated methanol is also having kinetic isotope effect value of 1. So, ethane and deuterated ethane, this kinetic isotope effect value of 1 indicates that the CH bond dissociation energy is not the rate determining step, but the same thing is also true for methanol versus deuterated methanol, where once again the CH bond breaking is not the rate determining step. So, based on these studies, one can think of having two different classes of substrate. Class 1 substrate where hydrogen atom abstraction is rate determining, as you see in these 3 substrate, let us say those are studies, methane, acetonitrile and nitromethane. For class 2 substrate, binding rate is the rate determining step. Substrate binding with respect to the active site or active species, that is the intermediate Q, that diiron 4 plus with the bis-mioxo intermediate, that is the rate determining step, right. That is quite fascinating, I would say, and this is true for C2H6 and methanol. Okay. One can perhaps think of studying many other substrates as well, but therefore overall the whole spectrum can be split into two. One is substrate 1, category 1 and the category 2. But I think you have seen the power of these reactions or power of this active site, and that is this bis-mioxo species are capable of forming the great methane to methanol. Okay. I think you have seen that there is similarity between the iron chemistry and copper chemistry. I will take that reaction mechanism in a moment, where you will see that iron chemistry and copper chemistry are similar. So, let me divide. So, let me try to draw the copper chemistry. What you have seen in case of copper chemistry, you have seen that copper oxygen or superoxo species is forming from copper 1, right. So, this is I am drawing copper 2. Okay. So, you have taken copper 1, reacted it with oxygen, right. So, one electron transfer happens to give you that, and from there on you have seen another copper can come in, copper 1 can come in to give you copper 2 O, let us say copper 2. I am deliberately drawing that as an end on, that will be easier to follow, and from there on you see the one electron breaking from there and one electron from there, another electron from there will give rise to the copper 3 intermediate, right, copper 3 bis-me-oxo intermediate. So, this is oxygen 2 minus, this is oxygen 2 minus, right. So, similar thing you have seen in case of iron, iron 2 plus, let us say, let me draw the similar intermediate, iron 3 would be there, similar looking intermediate if we are trying to draw iron 2 is reacting with oxygen, right. So, if we start from copper 1, we start from iron 2, it is a iron 3 superoxo, and from there on another iron 2 can come in, although this could be a side on geometry, just I am drawing as an end on just to keep it clarity, iron 3 can form. Now, this iron 3 intermediate can then once again give one electron, break homolytically, and this can give one electron, this one electron, and this can give one electron. Overall you can form iron 4 bis-me-oxo species. Now, you do see the similarity. So, this is a copper 3 bis-me-oxo intermediate, over here you have a iron 4 bis-me-oxo intermediate, only difference is one electron. Okay. We start with copper 1, we start with iron 2. So, we go to copper 3, we go to iron 4, right, that is very simple. This is why you can see that these species are very reactive and can effectively do a very great chemistry, and these are of course can do many chemistry as you have seen over here. Here you can react methane to give methanol. There are copper enzyme also which is responsible for these methane monooxygenases. These are the chemistry and these are the studies which are ongoing. The detailed understanding are still not 100% clear. Okay. I will not divert too much right now, we will come back later with the summary between the copper and the oxygen, similarities and summaries between them in some classes later. Let me get back to where we were discussing, and that is these are the class 1 substrate and the class 2 substrate we have seen, and these are the two different types of substrate that we have seen over there, right. That is fantastic. Now, let us look at the overall program then. So, this is once again from Professor Liffert's note, where we see that the first case, if you are looking at methane, that is in the black here. So, relative free energy, if you are looking at the black one, where it is showing that this is not really the high energy, this is diffusion is not the rate limiting, of course this is energy demanding but not too energy demanding. But if you follow the black line is having the CH bond dissociation is as the rate limiting phase, and then the hydroxylation product is forming. In case of ethane, that is in red, you follow the red line here, red line is very up. So, this transition state where the diffusion barrier is quite high, on the other hand CH bond dissociation energy of course is energy demanding but not too very high. So, this energy is much higher than this one. So, that is why this diffusion is the rate limiting, the CH bond activation is not the rate limiting. In case of methanol, the blue line over here, once again you see the in case of methanol, which is a the class 2 substrate. If you have forgotten, methanol is the class 2 substrate. Here you see that diffusion is far more energy demanding, it is very high energy. Of course, CH bond activation is also demanding, but compared to the diffusion it is less energy demanding. So, diffusion is the controlling factor over there. In case of nitromethane, as you have seen, this nitromethane diffusion is not too much of higher energy, but this CH bond dissociation is high energy. So, as you can see over here, so this relative energy plot. So, let us say just for methane, if you are looking at that is the black line over there. So, this is the diffusion energy compared to methane, the CH bond activation energy is quite high, as you see over here. That is indicating that this is not going to be too much troublesome, but doing this CH bond activation, the black line if you are following, is going to be the most determining factor or the difficult factor. So, to conclude, for this methane monoxygenase, I think we are very lucky to have such a great enzyme, where you have non-heme di-iron center in the very high oxidation state, iron 4 in the bis-muoxo bound intermediate, which has similarities between the copper 3 dicopper 3 bis-muoxo intermediate, but more importantly it can convert methane, even methane, to methanol. All other substrates are, I would say, relatively easier, not all, but most of them are relatively easier. There are two class of substrate that you can study with respect to this methane monooxygenase. One is class 1 or substrate class 1, another is substrate class 2. Substrate class 1 is the one where we see that kinetic isotope effect value is more than 7, or significant kinetic isotope effect value is there. If kinetic isotope effect value is equal to 1, then that is the substrate class 2. Those are the one, let us say for example ethane, that is what we have seen, and in case of also methanol we see the similar cases. So, the class 2 substrates are also can be utilized for its hydroxylation chemistry, but the diffusion is controlling. You have seen stepwise how these bis-muoxo species is abstracting hydrogen atom from methane, and then how little rearrangement is happening, making the hydroxo in situ form ready for the rebound, and then subsequent chemistry is following. That is fascinating. We will come back with much more chemistry in the subsequent classes. I hope you are studying these in detail, and also we will summarize them to make it a little bit easy and compare contrast among the different enzymes, so that you can follow well in the exam. Thank you very much. See you in the next class. Bye bye.